major legume crops, just around some 10 major legume crops. So, pigeon pea, chickpea, moonbeam, woodbean, then coffee, then lentil, rajmas. So these are the major legumes what we focus on. They are major insect pest problems. And so basically today's lecture I am going to cover the topic in these 6 areas. What are the major insect pest of legumes? All you might have been gone through in your MSc PhD. So basically I think you are from MSc and PhD or only MSc. Both are there. Okay. So accordingly I can give because you are well aware of the major insect pest of the legumes. So just I will give a glimpse. Then what are the microbial biopesticides? How it has originated years back and what formulations available for the microbial biopesticides and what is the global status and status of India in biopesticides. Then a small intro about the major biocontrol agents which we are using for legume pest management and what is the status at IITR. So this is the gist what I am going to cover in the next 15 to 20 minutes. So coming to the first, I have told PGNP. So PGNP, we are dividing the insect pest which first into 3 major divisions. Pod borer complex, you all know, it is a inclusion of gram pod borer, helicoverpa, then spotted pod borer, then blue moth, blue butterfly, whatever you tell, that which infers to the PGNP pods. Pod fly, which is emerging as a major threat throughout India. Then pod bugs, distributable septic. So this is the case of PGNP. Then we come to the Vigna crop. Vigna crop here, it's the moonbeam, woodbean, cowpea, then rice bean. This is the major legume which we cultivate in India. So in this, again, we have a list of insect pests like pod borer, spotted pod borer, then blue butterflies, pod bugs, in blister beetle. These are the major key pests which are emerging throughout the India. Then chickpea. In chickpea, the major pest is gram pod borer. Then apart from that, packets, cutworm is there. Then termites are there. Then in some occasional incidents, Spodoptera is infesting. Then lentil and field pea. So these 2 are North Indian crops, very little in southern India will be cultivating. So here again, you can see gram pod borer. So cutworm, then in case of field pea, leaf miner is a major pest apart from the gram pod borer. So then, as I had told, a little introduction about the genesis of the microbial biopesticides. Way back in 1834, talk about today's biological control. It was started way back in 1834 by Agostino Bassi, who discovered the first muscardine disease, that is caused by Bavaria, which you might have read in your UG course. So from that is the starting point for the liberal microbial biopesticides. Okay. Then just for academic purpose, I have given the definition. What is biological control? Be back. So it is given here. Then coming to the why we go for microbial biopesticides. Main advantage is that it is more target specific and safe to the environment and safe to the non-target organisms, especially the parasitoids or pollinators. And moreover, nowadays concern about environment, eco-friendly environment issue. So this is the main driving factor for the microbial biopesticide market. Then, when we look into the microbial biopesticides, the majority of the microbial biopesticide was from the first pie chart. You can see it's 64% of the microbial biopesticide is dominated by fungal, especially the trichoderma, then Bavaria, metarhizium. Then second major is bacterial. When we further look into the bacterium, majority is in Pseudomonas, the second pie chart. Then little is on 27% it is from Bacillus. So when we consider what is formulation, why we need formulation for microbial biopesticide? Literally, when we go for the microbial biopesticide formulation, we have to consider 3 major factors. That is, what type of microbial agent we are going to use, and what is the manufacturing process which we are going to prepare the microbial biopesticide, and what is the application environment. Simply, I can tell the motive of the microbial biopesticide formulation is to preserve the microorganism so that the shelf life and the efficiency of the microorganism should be viable until it reaches the target insect. Second, while storing microbial formulations, microbial biocontrol agents, they are live or almost dormant. For example, Conidia of Metaraxium or Bavaria or Trichoderma, they need some minimal nutritional requirement that has to be given by the then ecology and biology of the biocontrol agent. For example, BT endospores we are using in them. So it needs very minimal nutritional requirement and what type of environment it needs. For example, in case of Conidia, they need minimum moisture at the time of germination. So that has to be provided by the formulation what we are making. So these three factors we have to consider very critically when we choose a formulation for a microbial pesticide. In normal insecticide, you can got n number of formulation. In case of liquid, you can get E, then suspension concentrate, then WG granules, then CG. So in dry formulations, dust, then you can get wettable powders also. So there are so many formulations availability there for the chemical insecticide. But in case of microbial biopesticide, we have very limited formulations. When we see the microbial biopesticide consumption, US ranking first in the biopesticide market, followed by Europe. Then third is the Asian countries. So developing countries are in the booming of the microbial biopesticides. This is shown by the data also. We see the Indian market also. The microbial biopesticide market is picking up. As I had told, there is a very limited microbial bioformulation. In US and all, microbial bioformulations we can get granules in build, but in still it is lacking. So this is the formulations from CABRC in India. When we see BT, suspension is available. Then you can see the second wettable powder. So this is the major two formulations in India, which is approved by CABRC. So when we come to the Bavaria Baciana, three formulations are available. One is aqueous suspension, second is suspension concentrate. This is somewhat better formulation than wettable powder. In sense, what I had explained, it makes the Conidia live. Nowadays, there is a concept of giving a liquid bioformulation which enhances the shelf life of the microbial biospecimen. The case of Bavaria. Then, when we go for the NPV, which is a major biocontrol agent, third major biocontrol agent, only aqueous suspension is available in case of NPVs. And NPV also, only two NPVs are approved very long back, only Helicoverpa NPV and Spodoptera NPV. Apart from this, there is no further formulation approved by CABRC. When we see the fungus, Verticillium licari, here also only two formulations are available. One is wettable powder and another is suspension concentrate. So why I am showing all these things? There is a very much gap available in the formulation of the microbial biopesticides. So this is a small introduction about the bacterial biocontrol agent, which are, as I have told, so many insect pests of the legumes. BT is very bad managing the farmer complex. You all know it targets so many orders of our insect family. So already it has reached the boom BT. It had went up to the transgenic crops. Different insecticidal genes has been used, and the mode of action also you might have read on everywhere. So it's just a glimpse. Then coming to the entomogenic fungi, for legume pest management, we are using Bavaria, Metaraxium for the lepidopteran pest and defoliators. For example, Bihari caterpillar and Spodoptera, we are using this one. Then Verticillium lecani for aphids, whitefly management, and Hirsutella thompsoni also. Then coming to MPV, when we see the entomogenic virus, only the Bacillogridae family have the commercial application. In the Bacillogridae, as I had already mentioned, only Helicoverpa NPV and Spodoptera NPV is currently available. Even there is a scope for other NPVs also. Still now, in for the recommendation of the pest management in legume crops, in chickpea, we are advising Helicoverpa NPV 250 larval equivalent. Apart from that, we can go for the Bavaria Baciana. In case of PGNP also, 4 Correct? Because here the foliage is bit higher. That's why large volume is required. Then apart from that, BT. Then in beginner crops against Podoptera lectura and PV is there for tobacco caterpillar. Then coming to the spotted podborer, Maruka ulcerata, which is a major pest throughout India from north to south and east to west. Here also we are recommending only BT and metarhizium. These are all the two major biopesticides. Even long back they had identified the MPV of Maruka ulcerata, but still there is no commercial formulation available for Maruka ulcerata MPV. This was a from Taiwan country. Even recently, some 5 years back, full genome of Maruka ulcerata NPV was documented from AV and BC, but still the commercial formulation is not available. These are the lacunas I had told. Coming to the last section, what is the stand of IAPR in solving the microbial biopesticide availability for legume pest? In basically, we had gone for a survey from 3 different agroecological regions of the UP, and we had rhizospheric soil samples and dead insects. From there, we had isolated around some 63 bacterial biocontrol agents. We had morphologically screened it, and we had stained it using the Kumasi blue stain, and we had identified the endoscope forming gram-positive bacilli, which is basically the BT. From this, again, we had conducted so many bioassays, and we had found around 12 promising strains. Among the 12, 2 promising strains which were effective against 3 polyphagous insects. One is Bihar caterpillar, Spinosaurus obliqua. This we had tested against 2 different ages. One is 10 days old number, another is 20. And another thing is the Olepar SCD, which is a Castellari caterpillar, and another thing is Heligor parmicule. So these 2 strains F8 and AK2 IAPR, that is very effective against these insect pests. And further, we had done the SIM analysis also, and we had characterized the LC50 for different insects. For example, Helicorpa, Spodoptera, and Bihar, the Spinosaurus. Then, apart from this, the 5 promising strains was also done, and it is yet to be published. So, apart from that, for management of Bihar caterpillar, we have Spinosaurus obliqua NPV that we had isolated from episodic environment. So we had developed it as a formulation, and apart from that, we had documented one entomopathogenic fungi, Entomophaga albugi, group. So this we had documented. So again, BT I had already told it was very effective against the Bihar caterpillar. So this was the same analysis of the MPV what we had isolated, which is found to be a multiple nucleocapsid virus. So Spinosaurus obliqua, MNPV. So when we had analyzed the 4 genes of the there are 22 core genes. Able to identify it belongs to the alpha baculovirus group 1, which is very closely related to Hyphantacuna NPV. Hyphantacuna is a fall worm. So the road ahead for the biopesticides, the microbial biopesticide formulation is, one thing is that we have to look into the environmental stability of the biocontrol agent and the formulation, and the efficiency of the kill. And the third thing is the shelf life. So I thank one and all for patient listening, and I thank the team, SPGS, and Department of Entomology, and our sir, Dr. SPK, for giving me this opportunity. So I thank one and all. Thank you.
Whether the recipients of the same asset or after seeing the students of failure. The time allotted is not an issue. We come up with questions.
Yes, sir. We can do it. It should be 2 ways.
People who are working in process or biocontrol, And what are your microbial pros and cons? And you can also you are one thing normal is you will be attending biodiversity. In the course you will be having the biodiversity question there, when the first teacher will be asking something. Now it's your turn, I believe, right? You take it as your student and you can conduct biodiversity.
Feel free, it's just a learning, skill learning. From 2003, I have skill learning. It's still learning. So
what happens sometimes is answering is equal. Happening is
question is really
it's time to begin ask questions. Out of experience, I can ask questions and I can prolong. Prolonging is something, killing is some of the time.
you can ask
Better
people.
if it is come from you.
At least it will be useful for you if you are asking the questions.
And you can invite questions like and students here, they are coming from
I think in cool situations, people may not ask questions. Once
fully understood.
what it has gone, I said, superficial. That means my theory is not good, so you have not got up anything. I think that should not be there. If that is the case, you can tell me, I can explain one more thing very slowly. Because I think I was bit speed. So somebody, if you are not able to catch anything, you can ask it. In commercial insecticide, you are telling only granule formulations available. But for the beta-rhizium anisotropy, there is any.
We need to work on better concept problems ash to this.
Okay.
Approximately percent inside the soil.
So which kind of formulation will be more effective? You are asking. Good question. So your answer I have given in that I think one when I have answered. So that was the basic. So while you had chosen the biocontrol agent, meta-rhizium, please say. So you had chosen your agent as a meta-rhizium anisotropy. So what you are going to make? The formulation will be having only comedia or hypa plus comedia. What is your plan?
Also, from the soil.
Okay. You are going to prepare formulation. What you are going to have only comedia because there is one instrument called microharvester, which will harvest only the comedia and it will give you the comedia meta. If your intention is only comedia, then the formulation type will be different. Use the secondary metabolites of the fungus along with the comedia, then the formulation type will be different. So that's why I have told already, while you are formulating a microbial biosystem, first you have to think what you are going to take. Your active ingredient, I mean, only comedia or secondary metabolite plus comedia or type A comedia plus secondary. So based on that, your type of formulation will be different. Okay. So another thing is that where you are going to apply. You have a soil application. So for soil application, as we are telling, in case of fungus, don't think granule will be more efficient because in granule there won't be any moisture won't be. Okay. So I think you have to plan for a soil drenching kind of formulation. So where the microbial biopesticide should be to my suggestion, it should be a vegetable powder or some kind of liquid formulation will be doing better than granule. Why? Because once it is applied to the soil, immediately the Conidia has to germinate. So it needs more moisture. When you go for a dry formulation, that moisture won't be available. Because the farmer may apply irrigation, may not apply irrigation. So this is where you have to think. So the basic purpose of this education is trying you to think. It was a very nice question. So you think in that angle, what is your white dwarf? White dwarf problem in groundnut as we do. So again, as we will, Velocirus grub you are going to target. Grub, so it will be in soil. So as we is a major problem in which crop you are targeting?
Brinjal.
Okay, brinjal. So again, it is a rainfed crop. So rainfed plus irrigation condition. but better you go for a liquid formulation because nowadays concept is microbial biopesticide liquid bioformulations are picking up more because those they are having the longer shelf life rather than the dry formulation.
It is mostly talking about the level of plants.
Yes, right. Ah,
In the and
soda.
and as you rightly pointed out, is there any connecting in the laboratories equal or immunity to that extent? How about the case of collaterals?
In case of polyarthran, I will take the case of root grubs, especially in hurricane and sugarcane. What they had done in case of hurricane, there is a EPN formulation they had developed two different formulations from NBAR and Dr. Jagdish Patel they are there. So they had developed an EPN formulation. Um, it has a very good effect against the root grubs in algal. Similarly, in case of sugarcane, I think in sugarcane they had come up with a specific BT. Recently, they had done the full genome sequencing. Doctor, I think Srinivasan or from SBA, Coimbatore. They have done it, and they are into the development of a formulation that BT is specifically targeting a polio. Try3, having try3 gene.
So we can monitor this thing regularly.
Right. So these two cases I like to quote as an example for the polyamorphic management in the microbial information. One is EPN and another is the BT. These two are in trend. Even EPN they had shared the patent to three more firms in Karnataka. They are mass producing it.
How about government? Now, students take how about government? Government. Government not have to make a press. Clear plans. Clear plans. What are the basically big fish making? Renaissance making. And lots and lots to try to improve the populations are because most of the time we reckon that gone back 3 decades back because of this when we better than once a year came up recommendation. But what we hear from farmers point of view is not working here, but and it is mostly difference in this. Most of the places we are stories like it's not working. Which is this bicontrol also.
No,
Environmental factors. Very good. Environmental factors common. Like if last 18 pages you will say irrigate once in a week, spraying of pesticides, will not tell what pesticide it is, what dose will not. Environmental chemistry that is fine. Apart from that, it is not we meet with this failure.
visited a pesticide market, pesticide shop. There are so many products are available for the, especially the microbial biopesticide. That's they will show you a box, so and so. Whether graduate, postgraduate, you should have tested it. Whether really what they are showing in that box, it is there or spurious products.
That is one of the reasons. And what you said, environmental conditions, especially for fungus that works good. Fungus needs to be delivered at a low temperature, the temperature to be low, that way. And apart from that,
One severe thing is that no monitoring agency now for the microbial biopesticide quality assurance lab is lacking. Only the pesticide testing labs are there, but they are focusing only on the chemical insecticide. I think so. Very few care is taken for microbial biopesticide.
again, whatever is happening is last.
Ah,
Only from farmers' point of view, everybody's point of view. Things need to be fast. So, take up your microbial agents, most of the time they are
7 days.
not slow, faster, very faster. Slow.
Fungus especially 7 days.
And the government doesn't need that much time. You want to get results. That is the effect we get in. Because of that, what effect we say? Knockdown effect. So, Ratina, I remember how many fellowship were asked. Okay, Aparna, in the fellowship, we got fellowship. Similar. But, and one more thing, when I started fellowship, we are 2 years after fellowship for joining each other. Am I right? That's maybe 16 times scenario. I love the scenario. That is because of yeah. No, I said it was I said that you are sport the themselves come up with the fellowship. Here now I will choose the reading to criteria because 2 days back last week I attended a workshop here and the lecturer guiding us telling the thing is document here now also. We're getting so many help from other institutes whether if we secure admission in the fellowship assured like that. Presence but then you said that oh you might know the whole thing here. Start at the start it was not advertised. The lot of it was in a broader magnitude. How the rest must have because you don't see much of this failures in the theory programs. At one part of time, we started seeing failures in both work. Failure, we are supposed to be interpreted in that angle. But to period, only fail. Repeat that because we are fellowship for all the people. People, I mean, the way of taking, I'm in the kitchen, I will start like that. See, it is, what is happening in the cities, perhaps, you have no. This is one place where
They will get the fellowship for the MSc.
what they have done is, I mean, why they are thinking like this, probably this could be one of the reasons. They have more students. Last year, providing fellowship, the strength has been reduced.
Okay.
Fellowship. Then before that, it was 20, 25, there was something that we should be knowing. Just making a known explanation. Seriously, 22. Put together, when I in April when I left here, 120 or something, all put together, yeah. Yeah, it's still the money. Come on, huh? Yeah. Yeah, total of 120,000 in the average. Sometimes the trees in the back, it look like easy. That's the thing.
I remember when we are leaving, 9, 10, it was like that. Okay, very. It's just PG students are more.
When we left, it was only 5 or 7.
2000.
Got in place later. Your time, 5, 7, section, and it reduced to 10. But that make it other pages are not available to keep a register and somehow under the this committee, it is not permitted to treat something like that. There is no plenty of restriction that angle also. With the admission here, that is the thing.
Hey, exam is a great concept.
Well, getting into the thing.
MSc.
Not for membership.
Okay.
And that's why I'm asking them to keep up questions. So they will be answering so many questions, post maybe, most interest. So how about that one student got senior fellowship second rank, bonus in German fourth. Terrible. From Andhra, you see that? Yeah, she has gone by the mission.
It's higher.
Tulsi, policy, Tulsi, from Andhra Pradesh, to second rank. Canada, Andhra. So, status of things, that control which are slow, that can be attacked, other reasons here, other developments. And what is the isolate concept? Isolate concept. And what will you do for isolates? Whenever we talk of this fungus, most of the time we say isolates. And what do you mean by isolate? The same question, if it is asked, if you see in a printed form, very easily you write so many things, I know. This is where we have to add it to the table.
I think people are very less interested in microbial. That's why they are very sluggish.
Not so.
This was the stage where the NPV was in boom. Our TNE was leading the NPV. Even now, the status is in India, NPV nobody is carrying. Even the foam which was produced NPV, PCI, they had settled down so much. They are now producing very less amount of NPV.
And it's a division. It's some of the problems.
NPV.
I don't know.
Hyderabad. Dr. Vimala Devi, BT.
Vimala Devi.
BT. And now have only the established BT strain. Dr. Vimala Devi, Madam, BT strain was the first strain to be registered by CIBRC.
Not good support.
Now she had done the combination of BT plus Metarhizium. They had ordered now Doraemon side is picking up them. He is in a Yes, exactly. Now he is following up. We had a talk last week also. So they are into the now CIBRC has given approval for the combination microbial combining bacteria plus fungus.
Also, this is where going on.
So now dimension is open for making microbial products with combination products. Bacteria.
Why we are doing all this? The start working, you can live it now. But not working now. There. என்ன சொல்லலாம் தமிழ்ல சொல்ல ஹிந்தியில என்ன சொல்லலாம் கணக்குல என்ன சொல்லலாம் மலையாளத்துல என்ன சொல்லலாம் இல்லாட்டி ஒரு கொஞ்சம் இருக்கலாம் இல்ல trying to make it available or viable to all situation. போய் இல்லைனால ரெண்டாவது Now, when you like the paper now, look at the top of the city and you can think besides some of the things they close to you, right? Right? Because it can be viewed in some other angle. It showed beauty, the same flora, the same path, beauty and beauty. And apart from that, that's not that much. That's where my question is important. Why we are still in the completely can be we say they have instructors and they will come with its own things. It has to sustain. There was the answer. You cannot get along only with anchors alone. So something I will have to run. And every time you try to come up with the micro organism, the efficiency is going down. And that way you can learn to improve that. Improvisation of the existing ones. That's why we told us one more thing. Combinations. Consortium, so many things. Research topics, second years are here, now second years. Easy. The research program alone. And how do you think we are giving based on the presentation, or based on your chairman skill, or based on the student skill, or based on the student's marks are. The problem is anti-division of the programming. Once it is assigned, where it will end up, so not all the cases are not going to come up with a finding that is going to create wonders. It's not so. But something addition to the already existing things that may make it good. That's how we explain these standards. That's why the morning class also for the scenario when the presentation was made.
I was telling I was
So again, only mentioning again, non-issue not fire control, or because of yard in the house, so many are not under
You had asked a very nice question. So one of our colleague was also combining a domesticated product. We were asking only two. He was planning for five. First thing what we have done, first you have to do the compatibility test for the selected two organisms. Dual choice survey test they will tell and compatibility test. In your case, BT and Vivarium. So you try to inoculate different, as I was asking the previous question, nobody has answered. What is the difference between isolate and strain? Around minimum 50 strains of BT and 50 strains of Vivarium. Then cross combination should you have to test by the dual choice. So among which strain the more, and it should also have the highest efficiency. You have to look into those factors while taking a combination products. First thing is compatibility. Both should survive in a synergistic manner. It should not have an antagonistic. Generally, among the like it may not be there, but it is a mandatory. First, you have to check the compatibility of the selected two organisms. In our, I had told one example that our colleague he had tested 5 organisms, cross combination with all the 5. For example, he has taken bacteria, actinomycete, then fungus, then in fungus itself we have 2-3 different genus. So all the combination we had tested. Then he had found out only 4 organisms compatible. Then he had proceeded with that one. Getting a common criteria will be very difficult. In one, we have to go with the second of the biocontrol. That era going for only BT or only Metarizium and Metarizium conidia. As I was telling rightly, immediate action is needed by the farmer. Quick knockout. For that, you have to search for the so many naturally available secreted metabolites, secondary metabolites from the microorganisms. So for that, we have to do the basic survey. The niches where the natural existence of the host and pathogen is there. You have to look into those things. Then you have to isolate those kind of strains and study the metabolites. Then you have to go for efficiency study of that molecule. You can enhance the efficiency by enhancing the efficiency of the molecule by further fine tune. Those should be the criteria. So you have, in your case, you have to go for the and you have to select a media that sustains both. If you are planning for a just immediate aqueous suspension, then directly the broth converted into a compost.
PG, meaning of the patient.
Target insect. Maybe. No, you should target on order. Either level or focus should be there. Because when you go for that one only, you can cut down things accordingly. Order, at least order.
Only 2 are working. So how I started the future person checklist from the only 2 are working. We are actually concerned. We are not listening. We are working for
I'm working, sir, but I don't have it.
working with fermentation factors.
Lactoma, as I was telling, even though we are telling BT, even I had shown the some of the BT formulations. When I was planning for a review, I came up and with the shocking this one. Only till now we have only 2 indigenous BT strains of our own. What the commercially what we are using, it was imported from Russia, BT Kutstaki, imported strain. We should have identified this much strain, our own indigenous strain is. That was the main lacking. Indigenous. That was isolated from that Achaia Janata.
Even our own all legume, GDXLA, they are working now. It's not there in our process.
Formulations is not there. Only product was some packets it has occurred in that is that more. Formulations still it has not attacked the pulses. Fortunately.
I don't know.
No. If it is attacking, you will get some funds and projects.
Not a doctor will bring most of the times, always some disease to be always people to suffer. Not a policeman will bring. Because the kind of crimes only he can survive. Not a lawyer will. Somebody to kill somebody should do some legal and talk to movement, warrant should be there. Only the thief is interested in your reflect. If you are good, you can take something. That's how things go. Right? So,
It's very serious in Migla and PGMP also.
we don't have here, not much.
North India.
It is North Indian,
Especially in Figna, in Orissa, UP, Bihar, Madhya Pradesh. I mean also soybean.
you have your interview for that.
One aqua suspension for that one. That we got collected from the national episodic condition.
Suspension.
Aqua suspension.
Normal formulation.
Normal formation. As of it is the current topic, what she had was people had already tried encapsulation of BT, and this work was carried out in as I was telling in Microbial lab. They have started doing that one encapsulation, and even we have tried micro encapsulation, not nano encapsulation. That you can do. Where we can focus this kind of encapsulation means if we want environment. Suppose in sunny UV areas we are targeting, it will definitely increase the efficiency of the product to have more persistent. So it's people are started working in that area, but still level no product has come. So it's area to choose anybody in research on this encapsulation. It is in essence stage encapsulation.
Yeah, nanotechnology, that is fine.
Micro encapsulation, nano encapsulation.
Biotechnology.
Biotechnology, they have gone. Yes. That actually we have worked on the BT PGNP and BT chickpea from the National Agricultural Science Fund. We have gone up to the BR3, but research level 3 here. That is the even selection trial we have conducted. So we have a combination of Tri1 AABC and Tri2 AB gene in pigeon. It was a public funded into the PGNP. So I was associated in the transgenic. Stable transgenic PGNP and chickpea. Already trial was affected up to the even selection trial. State. DAC has told to clear the chickpea. Chickpea is final. In PGNP they had a small for the stability. So they had to give a told IAPR to have some homozygous lines. So that work is going on. For chickpea is already ready, but many scientifically advanced areas it needs government policy interventions. So as you all know, policy is not favoring GM. Only the last case recently they had. So if their government is ready for allowing GM crops, then first will be this, as you know, people's it will be ready. So in IAPR already we had worked out and we had found out with the lines. We have the lines of Tri1 AABC and Tri2 AB for PGNP and